Stage 1. Male cones and female cones are formed on the adult tree or the spore-bearing plant. Stage 2. Several male cones are formed in a cluster beneath a new shoot. 3. A longitudinal section of a male cone that shows it is made up of many fertile scales spirally arranged around a central stalk.
Stage 4. A longitudinal section through one fertile scale will reveal the contents of a pollen sac. There are two pollen sacs side by side on a fertile scale. Special cells called sporocytes inside the pollen sac undergo reproduction, division, meiosis to form many small spores or microspores. 4 microspores are formed from each sporocyte. Stage 5. The single-celled microspores still inside the pollen sac begin developing into pollen grains while still attached to their fellow microspores. A thick wall forms around each developing pollen grain, and 2 zones of the wall bulge out and become hollow air-filled sacs. Stage 6. As they develop, the microspores separate. Each microspore is a tiny single-celled haploid plant genetically different from the parent plant.
Stage 7. When a mature, each microscope germinates. This means that it begins cell division. By the time of dispersal, each pollen grain contains 4 cells inside a tough outer wall. These are the male gamete-forming plants, which are released from parent sporophyte. The pollen grains are supported by air currents to reach the female cones of another tree.
Stage 8. At the same time as the pollen cones form, female cones are formed near the tips of the branches, often protected by surrounding leaves.
Stage 9. A longitudinal section of a female cone shows that it is made up of many fertile scales arranged spirally around the central stalk. At time of pollination, the scales are slightly separated to allow pollen to fall between them to reach the ovules.
Stage 10. Each fertile scale has 2 ovules on its upper surface. Every ovule has a small opening in its integument, called the micropyle. Pollen grains are captured in watery droplets exuded by each ovule. The tiny air-filled sacs enable them to float up through the micropyle into the ovule to reach the surface of the female sporangium.
Stage 11. This shows a fertile scale cut longitudinally through one of the ovules. The ovule consists of the sporangium enclosed in an outer protective layer called the integument. A pollen grain can be seen on the exposed surface of the sporangium. At the center of the sporangium is one special cell called a sporocyte.
Stage 12. The sporocyte divides into 4 large haploid spores called megaspores. By reduction division, meiosis. Only one of the megaspores will remain functional. The other 3 abort and shrivel away.
Stage 13. The functional megaspore now germinates to form a female gametophyte contained in the ovule. The female gametophyte enlarges by multiple divisions of the nuclei, and the ovule enlarges to accommodate it. When it has attained full size, the nuclei form cell walls. Eventually, the female gametophyte is a multicellular structure several millimeters long inside the ovule.
Stage 14. One of the cells inside the pollen grain divides into 2 sperm cells which travel along the pollen tube to reach an egg. Stage 15. While lodged inside the ovule, the pollen grain has been growing a pollen tube through the sporangium tissue towards the egg-containing structures. 2 egg-bearing structures, archegonia, form at one end of the female gametophyte. A fully mature archegonium contains 1 large egg cell, as in Archegonium A. When a pollen tube reaches an archegonium, it releases the 2 sperm nuclei into the egg. The first sperm nucleus to reach the egg will fertilize it. The fusion nucleus or zygote is the first cell of the next diploid spore-bearing plant. Stage 16. 2 pollen grains entered the ovule at pollination. Both archegonia may be fertilized. Stages 16 and 17 show 4 successive stages of development, occurring in separate archegonia. The zygote divides into several nuclei, which all move to the opposite end of the archegonia.
Stage 17. The N2 will become 4 embryo cells. The cells behind them begin to elongate and push the embryo cells out of the archegonia. Towards the middle of the female neophyte, the elongating cells are called suspensors. As the 4 embryo cells are pushed deeper into the neophyte, they split apart into 4 separate embryos with the tips. Stage 18. Once the elongating cells have completed their task, they then the dividing embryo enlarges and forms a root tip, a shoot tip, and several seed leaves. By now, the surface tissue of the former ovule has hardened to form a seed coat, and the neophyte tissue is back to store nourishment. Then they arrange themselves into 4 tiers of 4 cells. Stage 19. While the ovules have been developing into seeds, the entire female colon has become large and big, with its scales tightly closed to protect the seeds inside of 4 separate suspensors. 4 embryos start to divide and eventually become multicellular. If 2 archegonia and fertilize this means 8 embryos are formed, only the most vigorous one will survive.
Stage 20. When a fully mature cone opens to release the seeds, if both ovules were pollinated, there will be 2 seeds formed on each scale. Stage 21. Attached to each seed coat is a papery wing, which enables the seed to be carried away from the parent tree by the wind. Stage 22. Upon germination, a seed gives rise to a seedling with roots, stem, and cotyledons.